want to do this?
Do we got all the technology?
He'll be your slide person.
Well, I want to welcome you all here, and thank you for having me. My name is Hal Hoffman. I'm a pediatric immunologist, and I live in San Diego. I work at the University of California and Rady Children's Hospital. I've been involved in the autoinflammatory diseases since we had the name autoinflammation. So I've had a lot of experience treating with a lot of different patients. What I want to talk to you about today, and I'd like this to be as much of a discussion and open-ended question as much as possible, is a little bit about some of the issues with genetic testing. So I guess my first question to you is, how many in this group, just by a show of hands, have either had your genetic test done or one of your family members' testing done? Okay, so a good number of you. Things have definitely changed over the last 20 years since we first identified the NLRP3 gene, which leads to CAPS and everything else. So a lot has really changed in the last 20 years. It used to be the only way you could really get a genetic test was by being involved in some sort of research study. But nowadays things have really changed. What I want to talk to you about is sort of the different types of genetic testing that are occurring right now, and give you a little bit of an idea of how it works and how to think about it, and also a little bit about what some of the advantages and disadvantages of each of the different types of testing. I guess, how many of you guys have heard about whole genome sequencing? Okay, good. How about whole exome sequencing? Okay, we're going to go over all this, so if you haven't, it's all right. What about panel testing? Okay, well, those are the three main things that are going on right now. What I want to give you an idea of, depending on if you're a reader or a sports fan, kind of what the differences are. So, next slide. Seth. All right, so if you're a reader, and I'm not really much of a reading for fun person, but let's say you read War and Peace, which is considered one of these long books. If you were to read that from sort of cover to cover, it's a total of 3 million letters. Now, if you do a whole genome, or if you actually read the entire genome from the beginning to the end, we're talking about 3 billion bases, so 3 billion letters. So it's 1,000 times War and Peace. And so what everything is going towards now is this whole genome sequence. And I do think there's advantages, and I think that's where we're headed, but the amount of information that we're getting, you couldn't carry those amount of, on paper. So really it becomes a data issue. All right, if you want a whole exome, then it's 30 million base pairs. So we're still talking 10 times, or 10 copies of War and Peace. So it's a really big chunk. If we get to the gene panels, it's a little bit more sort of manageable in terms of how to think about it. It's like reading several chapters out of War and Peace. But that's sort of the order of magnitude for each of these different types of testing. All right, for those of you who are sports fans, anyone here a sports fan? All right. Doing a whole genome is like watching a football game or whatever sport you like, from the kickoff until all the way to the end, and paying attention to every single thing going on at that, within every single player and every single play, all the way through from the beginning to the end, every second of that. A whole exome is sort of like reading it in the paper the next day. You get sort of some detail on some areas, but there's total areas that are completely ignored, and sort of the highlights. The panels is even more of sort of very focused. So what panels are giving you is really, they focus on a few players and sort of what those players did throughout the whole game. Generally, got that idea? All right, let's go to the next slide. That's where we're going to actually talk a little bit about what all that means from a genetic standpoint. Over here is the whole genome, that's called WGS. Over here is whole exome. then we have gene panels, and then we have actually the old school, which is called Sanger sequencing, which is sort of one by one, just looking at one by one different things. What happens when we are doing genetic sequencing is if you do whole genome sequencing, you are basically getting the letters, the bases, all the way from the beginning of the chromosome 1 all the way down to the chromosome 22, and then the sex chromosomes as well. If you do a whole exome, then all you do is just focus on the parts of the genome that we know code for genes. And so that's these orange sections right through here, because those are genes that we know that they code for a particular gene. If you do whole exome, then you basically, every orange part of your whole genome gets sequenced. If you do a gene panel, then it's just picking certain ones of those genes and certain ones of those exons. And if you do the Sanger sequencing, you're basically just doing either a whole gene or just sometimes just parts of those different genes. All right, how am I doing? Does anyone have questions? There's no question that's a bad question. Wow, do you guys really understand this? Because I get confused about this all the time. Seriously, don't feel bad about asking a question.
Do they use the Sanger sequencing in the whole genome sequencing? Is that a way that...
Not anymore. Oftentimes, the only thing they do with Sanger sequencing is it's still considered the gold standard. And although I think that may change in the future. And so oftentimes, if they find something in one of these ways, which are now called next-gen sequencing versus sort of single-gene or Sanger sequencing, they'll use this oftentimes to confirm these different things. The technology is for these three things is different than the technology for this. For the Sanger sequencing, what they do is they actually know exactly what things look like around these little orange things, and they can basically make many copies of this one little part of the gene, and then put that into a machine to give you the sequence. For these different technologies, there's this thing called next-gen sequencing, where they actually are carving up little pieces and then sequencing them and then lining them all up. And that's why you see here, this lines up enough sequence to sort of cover the entire thing. This really focuses just on the regions of where the orange, where the genes are, and then this just focuses on certain genes. Fair enough? I saw some other hands. Yeah.
You may be able to address this later, but are you going to be speaking about the different labs, for example?
Yeah.
And where those fall in?
Yeah, a little bit, but I'll need your help because I don't know if I know of all the labs. There's so many different ones, but we'll talk about that. And the problem is things change a lot, so I can tell you what I know at this time, but yeah, we'll get to that in the next couple slides. Yeah.
How do you feel about the future of the sequencing, given, as you said, it started with a data problem.
Right.
Now, as technology changes, to start getting different computing power and stuff like that, how do you see the future of sequencing genetics?
Well, one of the most amazing things that's happened, and really what's driven all this, is the costs have come down. It used to cost, when they did the first whole genome sequence, it was billions of dollars and took several years to do. And now they can do it for within $1,000 or $2,000 or $3,000 technologically, within sometimes 24 to 48 hours. So it's just amazing. The sort of problem is you get all that data and sort of how do you deal with it, and then do you know what it means? And that is also going to get better as we get better. Right now there's still, there's the technological component which can give you data, a lot of data, and we've gotten better at sort of dealing with big data. But there's still a requirement for people who actually can think about it to analyze it. Now AI, that's not auto-inflammatory, that's artificial intelligence, but AI is getting better and better, and pretty soon there will be a lot less human involvement in it. Now whether or not it's going to be as good, that's a big argument for a lot of different things. So honestly, where I think we're headed, and this has all kinds of issues for privacy and all kinds of issues for all kinds of things, is I think everyone's going to be sequenced. And it may be something that happens at birth, that that's the sort of thing that happens right when you're born, or it could be something that sort of happens, at certain times along your life, or anyone who sort of goes to the doctor. But I do see that happening in the future. Right now, it's still a little bit cost prohibitive, and there are still those privacy issues we've got to sort of learn to live with. But yeah, I think there's going to be many less barriers to having it done in the future, and we'll be better at sort of analyzing it. I guess what I want to say before I move on is that the sequence is only a small part of what actually happens. So you can, the DNA is that sort of code that sort of drives a lot of it, but there are a lot of things that aren't written in that DNA code. There are a lot of things that control what is expressed and whether or not parts of the different genome are expressed or what they do and where they're expressed. And so that can affect things. There's also a lot of things that happen environmentally that actually control what actually ends up happening with that genetic code. So it's not all about the DNA. And there are a lot of things we can't explain. Even if we've got the sequence there, it doesn't necessarily mean that you're going to be exactly like someone else with that exact same mutation. Fair enough? Okay. So in general, I don't need to sort of go through all these different things, but the concept is with Sanger, you're doing a few genes. With whole genome, you're doing 20,000 genes plus all the stuff in between where the genes are. They all sort of have some advantages and disadvantages in terms of the types of things that they can sort of pick up. And then they also have advantages and disadvantages in terms of how deep they can get, and that gets a little bit important when we start talking about somatic variants. And again, the costs here, I think, are relatively close to what's going on, although I do think that the whole genome sequence price is going to come down and down to the point where it's not prohibitive at all. All right, yeah?
Are you going to be addressing RNA sequencing methods? Just because I think it's important, so for my own son, he is technically diagnosed with systemic JIA, though we don't know, it might be as well called undiagnosed. We've tried all this, we've, tried the whole genome, whole exome, haven't found anything so far. And now we are working with the researchers to use more RNA-seq type methods, and that's being more successful. So I think it's important, especially for the patients with whom you don't find this, to be aware of those methods.
Okay. I hadn't put that on my list of things to do because it's not as available, but it's becoming a lot more available, and I think we can learn a lot about it. So what she is talking about is, so once the DNA makes a particular gene, so let's say gene 1 right here, what happens is then it gets transcribed into this type of molecule called RNA. And RNA basically gets rid of all the gray parts and splices it together. Now, it's complicated because sometimes it'll splice it in different ways. You can get lots of different forms. And again, even if it does get formed, sometimes that RNA doesn't get expressed. What RNA-seq is it's just basically sequencing of just the RNA. So it's just that sort of next level that happens after the sort of DNA is sort of made. And then once that RNA is made, that's what codes for the different proteins. And so every gene, sort of what it does is it makes a protein that then is either potentially affecting your immune system or affecting other parts of your cells. And so you can do a sequencing where they just look at either the sequence of the RNA, looking for potential mutations in that sequence, or sometimes the level of expression of that particular RNA. So it's at a totally different level, but I do think we're heading towards having that a lot more available in the future. Does that make sense? Okay, other questions? I'm going to move on. All right, next slide. Yeah, good. Okay, so as I said in the beginning, if the only way you used to be able to get sequencing is through some kind of a research lab, and a lot of times we were using Sanger sequencing, that sort of first type. But what's happened now is a lot of businesses have gotten into the business of doing genetic testing. So these are at least ones I know about. Has anyone had a commercial genetic test in a company that is not written here? Okay, well, got most of them. Oh, yeah.
I think we used Illumina, but is that the same as the mutate?
So interesting you said that. So Illumina is in San Diego. I know a fair amount about that. And they actually, they're what make most of the machines that actually these companies use. They are getting into the business of genetic sequencing. And so that is possible that they're also doing some of this as well. So Illumina is another possibility. It also could have just been the type of machine that was used that actually by different companies. Now, how many of you guys have heard about 23andMe and Ancestry.com? Right, that is not medical genetic testing. They can find sometimes information that could have medical relevance to you. They did a lot of studies that determined whether or not you can smell asparagus in your pee, or other things like that, which are super interesting. But they actually can also get you information medically. The problem is how they handle that data, and whether or not you're ready for it, and all those types of things. So in general, they are not designed for medical genetic testing. In general, they've used different technologies in the past where they just look for particular things, particular changes that we know about throughout the genome, and they'll give you information, and that's good if you want to know potentially where you're from your ancestry. And it may be good if you have a particular common change in your DNA that may change your chances of having a particular condition, but it's not really genetic testing from this standpoint, so you can't really use it as such. Question?
What about these panels of genetic testing that's showing you how drug interactions happen? I keep going to pharmacies and they say they can test.
Right. And they are using oftentimes a form of, sometimes they're doing panels, sometimes they're also doing some things like this where they know about particular changes that do affect the way you metabolize certain medications. And we are getting there. I think we're still not completely sure about that, but there are definite situations, particularly in the cancer world, where they use different cancer chemotherapeutics, and they know that certain ones are going to work better either for a particular cancer or are more likely to cause a side effect if you're on it. And so that technology, some of it is in the panels, and some of it is actually in some of the things more like this. So yeah, it's a good question. I think we're heading towards that, and that could potentially help us in autoinflammation, and we could learn that some patients may respond better to certain medicines, but we're not there yet. I think is where we're at. Okay. So all of these different companies oftentimes will offer panels, and the panels are very different depending on what they decide is important. Sometimes they'll offer for the autoinflammation a 7-gene panel, but they'll also, some of them will give you a 20-gene panel. And sort of what's decided in that autoinflammation is kind of random in that way. Some of them actually use others to sort of do their sequencing. They also report data a little bit differently. That last slide I'm going to talk about is a little bit about sort of reporting. And a lot of them are willing to give information a little to doctors or to the patients in a different way. And that sort of changes depending on laws and just depending on sort of the policies. So next slide. So for those of you that get a result back, the sort of standard classification of what that result means comes in these sort of 5 different varieties. So starting from the top, a pathogenic mutation has a pretty strict criteria in that it needs to be a mutation that's been described before, looks like it could cause a problem, and ideally has been proven in some particular other test to show that mutation actually does change the function of the protein. Lichen pathogenics are oftentimes, they have been described. There may be some data, but it's not absolutely sure. What a lot of things fall into is this thing called variant of unknown significance. Some people call them VUS or VUS or whatever you want to call it. This is one of the most challenging parts because these are situations where they could be a problem. They haven't been described before, or they have been described and they've been described in enough people that don't seem to have symptoms that we're not quite sure. And that doesn't mean it's not a disease-causing problem. It just means we just don't know. Then when you get to these are all of us, if you were to sequence our genome, are going to have lots of changes. I mean, we are 99.9%, 999% the same, but there are still lots of little changes that happen very commonly in the genome. And there are some things that we know cause no problem at all, and others that we're not quite sure. In terms of diseases, they do sort of change, things about us, no matter what, even if they're benign. All right, the other issue, it depends on which disease we're looking at, and which gene, is whether or not you have one or two variants in the gene. If you have a dominant disease, like CAPS and TRAPS or things like that, then all it usually takes is one mutation to actually have a problem. If you have a classically recessive disease, what we've sort of classically thought of is in order to have the disease, you need to have two variants. That's actually in question. And so there are a lot of situations where you may actually have mutations in one mutation in a particular gene that you're supposed to have two on, but maybe you don't need to have two in order to have problems. And so there's a lot we don't understand. Now, you also potentially, what we're finding is that not every disease is a single gene disease. And you may actually have a combination of variants that may not by themselves cause a problem, but in combination actually lead to a problem. So this makes us not fully understand a lot of different things, because in certain situations where someone may have a variant that we call a VUS, because there are many people that actually have that variant and haven't developed a problem, but maybe they have other variants and other genes that actually together with that variant lead to a problem. So we're still, even though you get definitive results on these testing, there's still a lot we don't know or understand. Sort of clear? Questions? And then I think I gotta stop. Yeah, go ahead.
So this is from birth, obviously. Is it
Not necessarily.
I was just about to ask, is
Great.
it possible to have,
Yes.
say, a chemical exposure or something? What can change this?
Yeah, it doesn't have to be a chemical exposure. I think that's we think about mutations from radiation or from chemical or things like that, but it is a natural process of our body to actually have what are called somatic mutations. And that can happen at lots of different periods of our development as inside the womb, all the way up until adulthood. And in a lot of ways, cancer is oftentimes one of these types of what are called somatic mutations that then cause a change in the cell that then can, from then on, lead to problems.
Okay.
So the other problem is when we usually do genetic testing, it's done by a couple different ways. One, it's either a blood test, or two, it's a spit test. And for the most part, all of our cells in our body are mostly the same, but they're not completely the same. And so there are some situations where all you need to have is a certain percentage of cells in a particular type of cell that have a mutation to actually lead to a disease. And so it may not be in your spit cells, or it may be in such a small percentage of your blood cells that it doesn't show up. So I'm glad you brought that up, because that was something I wanted to get to for sure. Is that, so the testing, even though you may come out negative, it may actually not give you the answer that you need. Go ahead.
Do tests like Invitae show the variant, the VU, well it's covered now, but the VUS or the benign, do they let the customer know? You know how you can go online and check?
Right. It usually requires the physician to request it, and it depends on the company. Some companies just have a policy where they don't get into it at all. Others have a little bit more of a lenient policy. If the physician says so, they're allowed to look at it. And so it depends on your doctor, but it may be that you need to sort of get your doctor to ask for that. Now the question is, we may get information and still not know what to do with it. And that's even someone like me and some of the other folks in this room who look at genetic sequence a lot. There's still a lot we don't understand. My personal feeling is you should have access to it. It needs to be explained in a way that doesn't scare you also. Because again, all of us have tons of different variants and we don't know what to do with those different things. And again, you can have a really, something that potentially could be really bad for someone and not be bad for you. So you need to be able to sort of have it in a way that's understandable, and know sort of that it doesn't, know sort of the pitfalls of sort of what that testing means, and not necessarily assume it's the one end to, going to tell you everything about it.
Thank you. We'll have some more questions a little later, okay?
Yeah, I know the questions doesn't matter.
Thank you.
Thank you. We also said we can submit questions via Facebook to our group, and then we can try and get them answered if there's not a lot of time here. Now we're going to have